anatomists and physiologists. This is continuing our urinary system anatomy series. In this section, we just finished talking about the structure of the nephron and what kinds of cells make up each portion of those tubules. And we talked about in the end of our last video how really the nephron loops back on itself. So we have that renal corpuscle, the glomerulus, those capillaries inside, the PCT, nephron loop, and then the DCT, the distal convoluted tubule, really loops back in between that afferent arteriole and the efferent arteriole. Because it's really like the last portion of the ascending limb of the nephron loop slash the first part of the DCT kind of loops back, that leads to some structural modifications. The area where these two structures meet is called the juxtaglomerular complex. Juxtaglomerular complex. Juxta means close to. Glomerular refers to those capillaries inside. All those capillaries are called glomerulus, and then complex is the combination of all the things. This is a really important meeting place because it regulates how much filtrate is formed at any one time, and it also monitors systemic blood pressure. So the juxtaglomerular complex, what it does is it regulates the amount of filtrate that's formed, and it also monitors systemic BP, blood pressure. There's two major cell types I want to talk about in the juxtaglomerular complex. I'm going to zoom in on this. So what we're looking at here is this is the renal corpuscle, that Lego hand structure I was telling you about. But then we have the afferent arteriole leading into that glomerulus. These are the capillaries, and they've taken like sections out of here so you can see like, oh, there's blood cells in there. All of the blood from that glomerulus leaves in the efferent arteriole. This is that portion of the distal convoluted tubule, the juxtaglomerular complex. The two major cell types, I'll highlight them in pink. The first type are macula densa cells. So the macula densa cells, these are the ones sitting right next to the glomerulus, all these little purple ones right here. They have chemoreceptors to keep an eye on sodium levels. So macula densa cells, they're dealing with NaCl levels. They're monitoring those because they have chemoreceptors there, and they want to keep an eye on what's in that filtrate. The other major cell type I want to talk about here are called granular cells. Granular cells are like modified smooth muscle cells, and they also secrete renin if needed. That's that hormone that can help regulate blood pressure. We'll circle back to renin when we get more into the physiology. But the granular cells, they aid in sensing the blood pressure of the afferent arteriole. So they're right next to this afferent arteriole. They can sense, these granular cells, the blood pressure going on in there. There's some mechanical receptors. These are some pictures that we're going to look at regarding the structure of the glomerulus. We're going to talk about that next. We're going to zoom in on each of these pictures. So we talked about the tube part of the nephron. I said we're going to circle back to the glomerulus, so here we are. Right now we're looking at the afferent arteriole. Blood goes into that glomerulus. Remember I told you it's like a ball of yarn, capillaries. And then that blood will exit via the efferent arteriole. The glomerulus lives in this structure called the renal corpuscle. That's this structure here. This whole thing, renal corpuscle. So the renal corpuscle is all of this and everything inside of it. Afferent arteriole is transporting in more blood and at a higher pressure than the efferent one, so that's why the afferent arteriole is larger in diameter. So remember that the glomerular capsule is kind of shaped like a Lego hand, right? It kind of wraps around these capillaries, but it also has a visceral layer and a parietal layer. So here's that parietal layer, and then there's also a visceral layer. The visceral layer of the glomerular capsule is just the covering of the glomerular capillaries. It also has some special cell types there that I'm going to highlight in pink called podocytes. Those are foot cells, which I think are so cute. They aid in making sure filtration happens in just the right way. So all of these like yellow bits around here, even with those little like finger-like projections, those are all bits of podocytes. Let's zoom in on that part. So right now we're looking at one little bit of the glomerular capillaries. Remember I told you that these capillaries are fenestrated, so there's some fenestrations or pores there. So those fenestrations are from the glomerulus, those ball of yarn capillaries. That means that these capillaries are pretty leaky. This is really great for producing tons of filtrate because we need that, right? But we also want to make sure that all the good stuff stays in the blood. And podocytes really help with that. And I think they're so cute. So the fenestrations, they allow for stuff in the blood to exit except for blood cells. So everything except blood cells exits. So all substances except blood cells leave as a result of the fenestrations. However, podocytes, they have these little things called foot processes. I'm going to highlight that in pink. Foot processes. So here's like the nucleus of that podocyte. Here's some of that's little extensions. And then off of those extensions are the foot processes. And you see how those foot processes kind of interlock in each other? I kind of think of foot processes how you like put your fingers, if you were to like put your fingers together and interlock them. You see how you'd have like little bits that can't quite like touch each other fully? That's like the foot processes. And they form slits, and I'm going to highlight this in pink too. They form these little slits, these little spaces called filtration slits. So there's little spaces in between where stuff can still come through. These little foot processes with the filtration slits act like a filter. Um, and so it's kind of the same thing. It's just a little tiny filter, and the filtration slits help make sure that macromolecules can't pass through. So prevent macromolecules from leaking through. Let me show you another picture of podocytes with the filtration slits. Here's another picture here. So here we have a big old podocyte. That's like the main part. And then we have those little extensions with those little finger-like, feet-like projections called the foot processes. And then those spaces in between, those are the filtration slits. So cute. What's not shown in the other pictures that we've seen is this little tiny layer in between the capillary and the podocytes. That little layer is this blue one here. So on this side is the wall of the glomerulus, the capillary, pink one. And then the yellow side is the little bits of the podocyte. What's in between is this thing called the basement membrane. I'll highlight that one in yellow. So the basement membrane, what the basement membrane is, it just regulates exactly what's leaving the blood to form the filtrate. And the thing about the basement membrane is that it helps block proteins, the large ones, which means that plasma proteins Plasma proteins, they stay in the blood. That's important, right? The plasma proteins stay in the blood because that's the thing that's contributing to our colloid osmotic pressure, the thing that draws water into the blood. We need that. So plasma proteins, we want them to stay in the blood. But also the basement membrane helps make sure that negatively charged stuff and other big stuff stays in the blood. The combination of these three layers is called the filtration membrane. So all three of these layers all together is called the filtration membrane. And that just makes sure that we're regulating exactly what's leaving the blood to form the filtrate. And we've talked about what stays in the blood, right? So let me write this down. Stays in the blood. The important stuff, right? Like blood cells, plasma proteins, negatively charged ions, macromolecules. What exits the blood is what's called filtrate. I just dropped my pen. So forms filtrate, which is the stuff leaving the blood, okay? This is going to be stuff like sodium, water, glucose, amino acids, which are not proteins, they're the monomers of proteins, but also nitrogenous wastes. Nitrogenous wastes. Just wastes that contain nitrogen, not something super fancy. And we'll continue to discuss this more as we get into the physiology. But the structure of the glomerulus and this filtration membrane that we're talking about really plays a role in how the filtrate is formed. We will talk more about this and I'll show you this picture again, but generally how the nephron works is three major processes. So from the blood, we'll enter that glomerulus from the afferent arteriole. We'll loop around in that glomerulus. And at the glomerulus, we perform the first thing, which is called glomerular filtration. So filtrate then leaves into the nephron. A lot of substances in that filtrate, they don't just stay in there. A lot of them get reabsorbed back into the blood. And that's called tubular reabsorption. So when stuff leaves the tubule back into the blood, that's called reabsorption. But some of the stuff from the blood is pushed into the nephron as we move through those tubules. And that process is called tubular secretion. So there's three major processes that happen. But all of these three work to form urine, which is the thing that's formed in the kidneys and the renal pelvis, which is then sent down the nephron, out the renal pyramid, the minor and major calyces, into the renal pelvis, down into the ureters, and then into the bladder. So thinking more macro, right? So from that collecting duct, which is why these renal pyramids look so stripy, we drain into that minor calyx, then into the major calyx, then into that renal pelvis, out the ureter, and then we move down onto the bladder. And how urine moves down into the bladder is through the process of peristalsis, contractions of smooth muscle in the wall of like all of this yellowy beigey stuff, including the renal pelvis and the ureter, all the way down into the bladder. That's what we'll look at next. So that urine moves down the ureters into the bladder. And here we're looking at the bladder and the urethra, in both male anatomy and female anatomy. The bladder is extremely collapsible, which means that when it's more empty, it's more squished and it's smaller, but it also means that it's able to be filled. So I'm going to zoom in on this one first on the female side. So where the ureters, so here's the ureter. Each of these are the ureters, and where the ureters enter are called the uteric orifices. Uteric refers to ureter, orifice is an opening. So urine is stored in here. Urine is stored in here. And then it exits out of the exit point, which is called the bladder neck. All of these form this kind of like triangular region. That triangular region has a special name. It's called the trigon. I'll highlight it in blue. So the trigon region. And the trigon region is clinically important because that's where a lot of infections kind of hang out, just because it's kind of like a flatter portion there. And there's layers of the wall of the bladder, which is much like the other hollow organs we've learned about. Internally, there's a mucosa. So the innermost layer consists of a mucosa. And then there's this muscle layer that's called detrusor. That muscle is called the detrusor. It's smooth muscle. And then on the outside, there's some connective tissue. The mucosa is made of this epithelium called transitional epithelium. Transitional epithelium, when the bladder is empty, it means that transitional epithelium kind of smooshes in on itself and it starts to form these folds. And they're folds that we've learned the name of before because those folds are called rugae. Those folds are called rugae. So all of these little foldy bits called rugae. But as that transitional epithelium and as the bladder stretches, that helps smooth out those rugae and makes the cells seem like pretty much columnar. And this helps the bladder be really stretchy without making too much pressure inside. The bladder, when it's full, on average, can hold about like half a liter or like 1 pint-ish of fluid. So then once we're ready to empty the bladder, that bladder will exit the urethra. The urethra. And this is controlled by two urethral sphincters, which I'll highlight in pink. We have the internal urethral sphincter, which on the female picture that we're looking at right now, female urinary anatomy, we have internal urethral sphincter here, which is smooth muscle. So internal urethral sphincter is smooth muscle. And then we also have the external urethral sphincter. The external urethral sphincter is skeletal muscle, which means that we have involuntary control over the internal urethral sphincter because it's smooth muscle. And skeletal muscle means that we have our external urethral sphincter is voluntarily controlled. The external urethral sphincter sits in kind of like this boundary point in between kind of like the insides and the outsides, which is called the urogenital diaphragm. And it also helps form the bottom part of the pelvic floor along with the other pelvic floor muscles, but we'll talk about that when we get to repro. The female urethra is quite short in comparison to the male urethra because the male urethra must travel from the bladder. There's that trigon region I was talking about. We'll travel through the internal urethral sphincter, but then we have to travel through all of the external male anatomy. And because the urethra in male anatomy is so much longer, that we have some distinct regions. So when the male urethra passes through the prostate gland, which is this yellowish ball-shaped structure here, that is called the prostatic urethra. When that urethra travels through the penis, that's composed of spongy erectile tissue, so we call it the spongy urethra. And then the part that's in between those, where it passes through the urogenital diaphragm, is called the intermediate part of the urethra. And then, of course, the exit point is called the external urethral orifice, both in male and female anatomy. That's what it's called. For males, that's at the end of the penis, and then for females, that's just at the end of um, the urethra. Both of those are at the end of the urethra. That, I think, is the end of this video, and we'll circle back to start talking about the physiology, and I look forward to doing our kidney dissection together. Woohoo!